Hi everyone, welcome to Biomix Hub. Today we are in the ninth part of the series of video in which we are going to discuss about some questions related to CSIR life science with their concepts. And today's main mode of discussion will be about the kDEL and KKXX sequencing. Now let us see the question. The first question is, the tetrapeptide kDEL is well known as a retrieval signal of several newly synthesized proteins. This process is mediated through specific receptor kDEL interaction. Any specific amino acid change in this tetrapeptide is not allowed in terms of its binding its receptor and its subsequent retination in specific organelle, whereas secretory proteins are devoid of such tetrapeptide. From this observation indicate the localization of receptor of this tetrapeptide. Here it is asking about the receptor localization of receptor where the receptors are present. Here the answer will be Golgi and ER both. Now let us understand the concept. Here we have two types of proteins in the ER. They are the resident protein, another one will be the export protein. Resident protein means their place is in the ER. They are stored in the ER. For further modification, they are not transferred to some other organelles. Next comes the export proteins. When we talk about the export proteins, they are the proteins which needs to be exported from the ER for further processing or for further modification. And the proteins which are present in the endoplasmic reticulum has two types of signals. They are the kDEL, EL and KKXX signal, and these signals are present in the C-terminal region. Now, what does the kDEL stands for? The kDEL stands for K stands for lysine, D stands for asparagine, E stands for glutamate, L stands for leucine. And in case of KKXX, K is lysine and XX can be any of the amino acid. Now, where this kDEL is present, where the kDEL and KKXX is present, it is present in the C-terminal region. Now, when we draw the diagram of endoplasmic reticulum, here suppose it is the endoplasmic reticulum. The proteins which are present inside the endoplasmic reticulum, inside the lumen of the endoplasmic reticulum, they will have the sequence of kDEL, where in the C-terminal region. There are some proteins which are present near the cytosolic phase in the transmembrane. Here some of the proteins will be present, and these proteins will have the signal sequence of KKXX in the C-terminal region. Okay. Now let us see some other things. Suppose it is the endoplasmic reticulum. It has some of the proteins. These are the resident proteins which are present in the lumen as well in the transmembrane side. What happens? Sometimes they bulge out. These proteins are exported outside. These proteins are exported outside. How? In the form of a vesicle. Whenever a protein is to be exported, it will be in the form of a vesicle, and it will be coming near the Golgi apparatus, and it will be received by the Golgi apparatus. This is the Golgi apparatus, and this is the endoplasmic reticulum. The proteins which are present in the lumen, they will be exported. The resident protein, when they are exported, they will be in the form of a vesicle, and the protein known as COP2. Here COP2 will be coated in the vesicle. then it will be transported to the Golgi apparatus, and the Golgi apparatus will receive this vesicle. This vesicle will be along with protein will be received by the Golgi apparatus. When the Golgi apparatus will see that it is having a kDEL or KKXX sequence in it, then what does Golgi apparatus will do? It will again transfer these proteins into the ER, and if it sees that it is not having the kDEL or KKXX signal sequence, it will modify it to go to the lysosome, or further degradation will take place. Further degradation or modification will take place. Now, when this protein is coming, it will act as a ligand. For binding this protein, there needs to be a receptor present in it. The Golgi apparatus is having a receptor which will bind this protein, which is having the kDEL sequence. Here it is when the Golgi apparatus will recognize the proteins which is having kDEL sequence, and it will bound with the receptor. Now, when it is bounded with the receptor, it will export again into the endoplasmic reticulum. It will be exported to the form of a vesicle. Here another vesicle will be formed in which the protein will be present. Now, the Golgi apparatus which form the vesicle, it will have a coat which is known as COP1, and it will transfer this protein in the form of a vesicle into the endoplasmic reticulum. So, now there is another concept. Will the ER have a receptor or not? Yes, the ER will also have a receptor within it, which is responsible for the binding of the kDEL sequence proteins, which are the proteins of endoplasmic reticulum. Still, sometimes it cannot bind because the endoplasmic reticulum has high pH. Its pH is high. When the pH is high, its binding affinity will be low. That is the reason the receptor of KD which can receive the signal sequence protein of kDEL, it cannot bind. But when it is transported into the Golgi apparatus, the Golgi apparatus will have low pH. pH will be low in case of Golgi apparatus. As the pH is low in Golgi apparatus, its binding affinity will be high. So, the proteins which is having the kDEL sequence will bind to the receptor of the Golgi apparatus, and again it will be exported to the endoplasmic reticulum. So, our question was that where these receptors are present, where the kDEL receptors are present. So, our answer will be it is present both in the ER as well as in the Golgi apparatus. See, we have both the options that it is present in Golgi apparatus as well as endoplasmic reticulum. Sometimes we pick the answer that is the third answer, that is endoplasmic reticulum, because kDEL is a protein of endoplasmic reticulum which is having this sequence. So, we can pick this answer, that is endoplasmic reticulum. But sometimes it is not received by the receptor of kDEL. So, Golgi apparatus is also having the receptor. So, our answer will be Golgi as well as ER. So, we have discussed one concept of CSIR life science question, and thank you for watching this video till the end. If you like this video, please don't forget to subscribe this channel and press the bell icon for further notification. Thank you.